One of the first questions that has come through is regarding for stability data that are published on in various journals and the ones that are available in stability.org database. So the question is, can data be used for generic drugs too? So perhaps maybe Connor, if you'd like to comment.
Yeah, yep. So when we tested flucloxacillin, we just used, we surveyed all the generic preparations that are available and just chose one. It wasn't any one in particular. So I think most generics, I know with piperacillin tazobactam, for example, they changed the branded product Tazocin. They changed the formulation slightly, which would be a problem in using that versus generic piperacillin tazobactam, I think. But generally speaking, most antibiotics don't have anything other than the antibiotic in the vial. There's some citrate, I think, sometimes, but yeah, so usually you can extrapolate across from one generic product to the other, as long as it's meeting the pharmacopoeial specification, which it would need to do to be licensed.
So yeah, yes, great, I completely agree. So the next question is from Mark is, so I think perhaps Conor, you did allude to this in your presentation towards the end. So the question is it ideal to lobby pharmaceutical companies or licensing agencies to undertake stability testing for all new antimicrobials, particularly around the gram-negative ones? So,
sorry, say that again, Paddy, was it?
So regarding the need to lobby pharmaceutical companies or
yes,
licensing agencies to do stability study as part of the product approval process.
Yeah, absolutely. And I think, we've had a conversation around that with one of the sort of major pharmaceutical industry partners. Just, start thinking about that. It would be so easy to do in the work that they're doing prior to submission of a licensing application. Rather than do it after the event and having to look for grant funding or other funding to do the work. Build it into the licensing process, and then you'd have the option to probably do lots of different temperatures and concentrations and have really good looking buffers and stuff prior to licensing, and I think that would be really helpful.
So the next question, perhaps Nicolette may comment on. It's regarding the citric buffer acquisition in Australia. So you've kind of answered it in the chat box, but I would like you to comment on it. And also, if I may add to this question. So I know that 4% citrate is available locally. So what strengths do you use in your clinical practice in your pediatric setting?
Thanks, Akadi. Yeah, we actually get back to pre-filled syringes as well, and it depends on which product we're compounding, what the final dilution is. Certainly, it's very similar, I think, to the product that Grace is referring to in the Teams chat or Zoom chat. So yeah, I guess finding out what your local compounder is able to supply. Certainly, we haven't used vials as such. The pre-filled syringes or the citrate bags seem to be more cost effective for usage.
Okay. So in line with these buffers for stabilizing agents, one of the challenges when we do stability studies is buffers at low concentration do have a low capacity in terms of maintaining stability. At high concentrations, we note, so they can stabilize the pH and the stability of the drug as well. So one question that comes from Jason is whether there should be any global agreement on the data or the strengths of buffers that are allowable to use in clinical practice. So maybe Connor, if you would like to comment on this one.
Yeah, and just to echo the point, I think Grace is maybe making on the chat about the challenges of finding these buffers. And we had a similar situation in the UK where we were able to get sort of a pre-prepared 0.3% citrate buffer, which in our hands seems to be the most useful strength that we've found for Piptas fluclox. And just as an aside, with reference to your point about the strength of the buffers, when we looked at for meropenem stability, we went up to a 5% citrate buffer, and still it wasn't particularly helpful. But meropenem is a really challenging drug at the best of times. I think it's really important to have some sort of international consensus and availability of buffers, because again, it's another barrier. So if you have to buy in a higher strength buffer and then dilute that, often the safest place to do that would be in the aseptic manufacturing facility. But if you've got a model where you're trying to prepare, and I know some places do this, prepare an elastomeric device near the patient at the patient's bedside, if you have to introduce 2 dilution steps, so reconstituting the antibiotic and then diluting it, and then with the buffer, which you also have to dilute, then that's introducing additional risk, I guess. So it feels uncomfortable. So I think being able to have a consensus on these are the buffers that are useful, and also having an understanding of their clinical impact, because we've looked at that. My colleague Tim Hills looked at that in at Nottingham University Hospitals to see if there was any sort of physiological impact with a lot of citrate. If you imagine a patient having an elastomeric device for maybe 14, 21 days, something like that, you know, what is the impact, and what monitoring do we need to do? So coupled with having the availability, it's really have to be really good to have an understanding of their clinical impact themselves on the patient, and if any additional monitoring is required.
Thanks, Connor. So the next question from Emma is regarding ceftazidime and pyridine toxicities. So the question is whether any monitoring was done, or if anyone has any experience clinical toxicity observed from pyridine. So Nicolette, do you have any experience on this one?
I'm smiling because this rocked our cystic fibrosis cohort a little bit when the information came out about this. And I remember when we went looking for the information, actually had a chat with our local toxicologist around this. There's very limited information about the impact in terms of the toxicity, and I can see Connor shaking his head, so hopefully that's the same experience in the UK. What we did in terms of the communication with this particular cohort of patients, because they were using so much ceftazidime, was just to say that, you know, these patients are closely followed up from a medical perspective. We are looking for side effects, and as new information becomes available, we would share that. I don't believe there's been anything published as such, but Connor, please correct me if I'm wrong. I certainly note that our change, we've actually moved completely away from using continuous infusions, even over 12 hours for ceftazidime. So we've got a hybrid model of care where we've got elastomeric devices that deliver the dose over half an hour. So our nursing team from the hospital home team would visit in the morning, give Tobra and Ceftaz the first dose, and then the other 2 infusers are connected by the family at home. And so we provide aseptic not touch technique and central line access training to them, and they are signed off before they go home. So that's a different model of care that we've done as a result of drug stability issues.
Yeah, just to echo, I think what Nicolette is saying, you know, we don't have any hard data on pyridine toxicity, and it's interesting to see the difference in approach. So the USP, I think, and the European Medicines Agency both limit pyridine to 2 milligrams daily, maximum daily allowance, whereas with the British Pharmacopoeia, they allow 0.5, not more than 0.5% of weight per weight of ceftazidime. So in our study, when we looked at it, we would suggest that you could have between 15 and 30 milligrams of pyridine and still be within the British Pharmacopoeia limit. And there was a really nice review, I think, that Terry Jones did, I think, looking at, you know, pyridine, and, you know, there's all sorts of dietary sources of pyridine. You find it in ice cream, you find it in strong coffee, bacon. So, you know, how you separate that out, that toxicological consideration from the medication versus the diet and the background exposure, I don't know, but we don't have any data. But, you know, that is a very hard limit from USP and the European Medicines Agency up at 2 milligrams. And, you know, without anything to say that it is safe, how can you not comply with that? So it's very challenging.
Okay, thanks.
But I would also say, even with ceftazidime, concentration is a challenge and temperature is a challenge. So even if we could resolve the pyridine issue, there'll still be lots of problems with trying to use it in over a 24-hour period. And as Nicolette says, you know, the short infusions seem to be the way to go, which is unfortunately not hugely convenient.
Thanks, Connor. So while we are on ceftazidime issue, there is another question directed to Nicolette, which says, so do you use ceftazidime 24-hour infusion with Colipax for OPAT, or still do you use 2 times 12-hourly infusion in your practice?
I'm smiling because I'm pretty sure this has come from my colleague down south. So no, I've not been able to convince our team, and to be honest, I agree with us using our modified HITH model. From a drug stability perspective, the dose of ceftazidime we utilise in our cystic fibrosis cohort is 100 milligram per kilo per dose, up to 4 grams per dose 8 hourly. So it's really big industrial doses. And we've not been able to find stability data that we felt comfortable with to then go back to our CF cohort to say we can provide this service with 12-hourly dosing. Cold packs, cold IV infusions are extremely uncomfortable, and if you're trying to convince a child to have that, that's probably not a good way to go. And when they have to have that for 14 days, which is our standard duration for cystic fibrosis pulmonary optimization, usually doesn't go well. So I have to say, I think the compliance or adherence with the 8-hourly dosing using the elastomeric devices with the hybrid model has been quite successful, and we haven't seen worse clinical outcomes as such with those patients. So I don't feel we've necessarily reduced efficacy as a result. I know that extended infusions would be a benefit if we were utilising this for gram-negative sepsis, for example, or gram-negative infection. But I think in our cystic fibrosis cohort, we're still achieving good clinical outcomes on the 8-hourly dosing.
Thanks, Nicolette. So the next question from Grace is regarding degradation products. So when performing stability studies, if unknown impurities are discovered on HPLC, is it a general practice to further investigate what the impurities are, or do we just leave them as they are? So perhaps maybe Connor, maybe you may comment to your experience from your stability study program.
Yeah, so unfortunately, we don't have the sort of the capacity to sort of pursue the degradation products and understand them other than just characterize them in the sense of their, say, separation on a HPLC trace, for example. So what we would tend to do is have a look at the, in the sort of pre-scoping work, we'd be looking at the literature to see what degradation products are already identified, and if they are anything considered to be or known to be harmful, and then look to see if there's any pharmacopoeial limits that would apply to those. But obviously you'll see lots of degradation products with potentially with some molecules, but we've not been able to sort of characterize them any further and look in any more detail other than to note that yes, there is degradation. But the HPLC process that we should use is for stability indicating is its principal aim, rather than sort of analyzing the degradants in great detail. And yeah, we did a lot of work and liaised with colleagues from the pharmaceutical industry with ceftolozane and tazobactam, see if there was any degradation products that we needed to be aware of, and couldn't find any. So we did quite an extensive search, and unfortunately there often isn't a huge amount of information in the literature, and the knowledge of the degradants is often held by the sort of pharmaceutical industry and the company that has developed that, so they're often a good source of information if there's any concerns.
Thanks, Connor. So the next question from Aziz is regarding the complications related to administering antibiotics by IV line at home, such as infections. So what is the clinical experience? So who wants to comment on this one?
Nicolette, you want to go first?
Nicolette, first.
Happy either way. Very brief. I think obviously IV complications such as thrombosis or tissuing of the catheter, thrombophlebitis, all of those things are definitely not something that we want as an outcome. Certainly from a line infection rate, I'd like to say that we are very fortunate the way our services run and the training is provided to staff as well as families who have to handle central lines or PICC lines. Our line infection rate is very low, and kudos to the team in providing such excellent care, because it's because of them that we do have that low infection rate. In terms of thrombosis, with our vascular access service, again, that I've mentioned before, have done a lot of research in this space, and Mark Gilchrist asked a question around midlines earlier. Interestingly, Mark, we have actually not experienced a very high rate of thrombosis in our midline group of patients, and I'm happy to share the study for those who are interested. Again, it was our cystic fibrosis cohort, and we were comparing it with PICC lines. And this was particularly around avoiding general anaesthetic. But certainly for some patients, there is an increased risk. So adolescents, previous lines, or other comorbidities such as liver disease that increases the risk of thrombose.
Connor, would you like to add on this?
Yeah, it's just not particularly my area, but from what I understand from looking at the literature, there seems to be very low, low rate of line complications in OPAT, and it's well regarded, I guess, in terms of that element of its safety. But yeah, I don't have any direct experience to add.
Okay, thanks. So we'll move on to the next question, again directed to Nicolette from Mark. So Mark is saying that they are starting their paid OPAT service with continuous infusion. So any tips when starting in this population, similar to the adults, regarding selection of and any situations to consider when pumps have failed?
I'm smiling because we've learned by trying and sometimes failing with a new device. But we're very fortunate. We've got a fantastic hospital and home nursing and pharmacy team who have really gone to find a device that would be suitable to use in a range of different patients, especially for continuous infusions. There are occasions when elastomeric devices are the best choice and provide good drug delivery, especially when the dose is within what we would call standard limits. But for infections such as, bone and joint or infective endocarditis, where we do push the boundaries in terms of dosing and we do give more or higher dosing, we do find that the elastomeric devices just don't perform as well. And so being able to compound in an IV bag and running it via what we call an ambit pump, which is completely battery operated and it still allows the patient to move freely and is quite safe to use. Plus, you've got the guarantee of having your drug dose delivered over 24 hours. That's been our way to go. And certainly there are lots of devices available that are ambulatory devices that may provide the same flexibility in the UK. But, this is something that we've found quite useful in Australia. And yeah, I have to say it's just about finding a good fit for your patient. And sometimes the IV access does determine what you get to choose. And probably the most important thing is, from an AMS perspective, try and stay away from those once daily third generation cephalosporins if you can avoid it.
Thanks, Nicolette. So the next two questions again relate to the citrate buffer. So the first comment from Grace is regarding the dilution procedures for citrate and being arbitrary. So if I may comment on this one, so I did try to estimate the final percentage of citrate for the different dilution procedures from one of the local hospitals in Queensland. So I agree with your comment, Grace, that the dilution procedures are a bit arbitrary and the final concentrations are not the same, but generally they range from 0.2 to 0.3% citrate in the final product. Yeah, but I think earlier it has been commented there is no such standard agreement on the percentage of the buffers, but the buffer concentrations are really very low as well. So perhaps this is something to consider for the future, maybe in a study as well, to harmonize the dilution procedures so that we have the same final concentration of the citrate buffer in the final product to ensure stability of the pH in the buffers. So the next question relates to the supply. So maybe I will direct this one to Nicolette. So do we have a supplier for the citrate general ampoules or packs? So they are, I think our attendees says that they are having a hard time finding them locally here.
Yeah, I think if the attendee is happy to perhaps just send a message to Luminita with their email address, we can provide some details. Obviously, it depends on what's available in your state, but certainly we can share where we've previously been able to gain access. And certainly Grace has provided some great comments around citrate buffers. So perhaps Grace can also share.
Okay, thanks. So we're almost exhausted with the questions from the participants. If there are any other questions on, one question just came. So can citrate be used as a stabilizer for all the antibiotics? Maybe Connor has a lot of experience with using citrates, so I would like to give this opportunity.
Yeah. So when we're looking at doing a stability study on a drug of interest, and our sort of list of drugs of interest has been sort of provided to us by our OPAT community as to what the ones they think would be the most useful for their practice and to help them extend their practice. So often if we've got a molecule that we think isn't going to, well, from the literature we know might be a stability challenge, we will look and try a range of buffers, so phosphate, citrate, citrate phosphate, we've looked at a whole variety. For us, it seems to be so far for penicillins that citrate is the most useful. So there's also, we haven't done the work, there's also published stability data for benzoyl penicillin using citrate as well. So 0.3% citrate for us seems to be it's kind of our go-to buffer, and we'll always think of that one first. But that's not to say that there might not be better buffers out there with better buffering capacity. We tend to look at that in pre-scoping, and then depending on the results that we get, we go with the one that looks like it's going to give us the longest fridge time and meet the longest possible infusion time, and ideally 24 hours.
Thanks, Connor. So I think we have now exhausted all the questions. So I would like to give opportunity for our attendees, if they would like to make any comments, maybe one or two comments on any of the points that I've been discussing. So you're most welcome. So maybe while waiting for that, maybe a final comment from Connor regarding the 5% versus 10% arguments. So do you think a PK/PD approach will be a solution for this one?
Yeah, I mean, I think it's really interesting, and I think it's something we really do have to look at, because Nicolette's mentioned already the challenge of gram negatives and stuff like that. We don't have a huge range of agents to choose from, and we do have some very difficult to treat infections, and we have a patient population who needs to have as much access to as many drugs as possible in all the various different formats, whether that's self-administered, continuous infusion, twice daily visits, whatever it happens to be. The stumbling block, I think, for me, is that pharmacopoeia standard. So if you're the pharmacist, maybe releasing these products and knowing that maybe they're going to be used for 24 hours and the losses might exceed the general pharmacopoeia limit of 5%, and also when you factor in that there's variation at the start of the concentration at the outset, so due to reconstitution errors or overage in the bag, or changes in the drug concentration with the freeze dry process during the manufacturing, or the losses of the agent during storage of the vial of antibiotic before you reconstitute that. All of those things, and the considerations of those are why in the UK the British pharmacopoeia limit is 5%. Appreciate that obviously the rest of the world is 10%, so for a lot of drugs. So I'm not quite sure how to square that circle personally, and it's maybe a conversation to sort of have with the regulatory authorities as to what they would be. But of course, the one thing I would say is that we're clear as well that if you've got evidence that you're meeting your PK/PD targets, there's no particular concerns about any toxic degradants, and you can be assured with good critical review of the evidence that a 10% loss would be acceptable. It's not an absolute barrier if you do lose up to above 5%, if you do lose more than 5%, but it's kind of on a case-by-case basis, I guess, in terms of the antibiotic and what we know about it. But it's not going to be a conversation that ends quickly, I think, if I'm honest. I think it'll take quite a bit of discussion around what we're comfortable with clinically, from a regulatory perspective, but as I say, I think bringing the PK/PD argument and the modelling of total exposure into the discussion would be really interesting.
Thanks, Connor. So we have a final question that has come through. So regarding the different strategies for stabilization of antibiotics in addition to pH-dependent optimization, for example, the possibility of using novel formulation. If I may quickly comment on this one. Yes, as a part of our drug stability testing program, and also for OPAT drug optimization involves looking into the use of novel formulations based on nanoparticles to stabilize some of the drugs. So we have some preliminary experience with meropenem in collaboration with the School of Pharmacy here at UQ. So yes, I agree totally with the comment that novel formulations are potentially a solution for some of the very challenging antibiotics, such as the carbapenems. I would like to thank everyone for your patience and for those of you who have stayed this far. And I would like to give special thanks to Dr. Honor Jamison and as well as to Ms. Nicolette Cram for their interesting presentation. Thank you very much all. So I hope we will see you again in another session on OPAT and related topics. And thank you very much.
Thanks, Ricardo. Thanks, everyone.
Good night, everyone.
Thank you.
Bye-bye.